Hi, and welcome to Mr. Fisher's Biology Podcast Series. Our next podcast is on enzymes. So what are enzymes? Well, the first thing enzymes are, for the most part, are proteins. And remember, proteins are made of amino acids, and that's one of their functions. Enzymes are also catalysts. And just by definition, a catalyst is something that will accelerate a reaction without being permanently changed. So enzymes can be reused in reactions. Enzymes are selective, meaning that enzymes will only work on the substrates that they're specified for. And this has to do with their 3D shape. And finally, a useful way to remember enzymes is they all have an ASE ending. So some examples would be enzymes like sucrase, and sucrase is an enzyme for the sugar sucrose, maltase, which is an enzyme for the sugar maltose, and lactase, which is lactose. So enzymes, how do they work? Well, an enzyme has a very important role in our body, and what they do is they lower the free energy of activation. So if we look at this graph here on free energy, without an enzyme, we would need to add this much energy here in order to allow a reaction to happen. So in our bodies, this would mean that we would need a lot more energy, which means we would need to eat a lot more foods in order to allow reactions to take place. This wouldn't be very good in terms of homeostasis and allowing an organism to survive. What enzymes do is they lower that energy. So here's how much energy would be required with the help of an enzyme. So enzymes are catalysts in that they speed up a reaction. They allow less energy to be used in order for a reaction to take place. This is a very useful graph to explain the purposes of enzymes. So just a little bit on terminology. The substrate is the substance that the reactant or the substance that an enzyme acts on. So if this is our enzyme here, the substrate is right here. I like to think of these and as Pac-Man. And the Pac-Man being our enzyme, and the little balls that the Pac-Man ate would be the substrates. An active site, that would be our Pac-Man's mouth. And that's the region where the enzyme will actually bind to the substrate. Okay, now there's the concept of induced fit. So when the substrate interacts with the active site, the enzyme changes its shape. And this is largely due to hydrogen and ionic bonds. And so we can imagine, going back to our Pac-Man analogy, that when the Pac-Man comes near the little ball, its mouth closes and it eats it. In a way, that's what happens. If we look at an enzymatic reaction, we can see that we have our substrate, and we're going to use sucrose as an example. So we have a substrate, could be lactose, could be maltose, could be any other molecule in our body. We have the enzyme. The enzyme is, in this case for sucrose, is sucrase. So when those two interact, they create what's called the enzyme substrate complex. The enzyme substrate complex is then able to break the sucrose into its products, which are glucose and fructose, into its monomers. The end products leave the enzyme intact, so the enzyme is able to work again on another substrate. And that way our bodies don't need to keep making a new enzyme for every reactant that we need to break down. So there are some things that affect enzyme activity. We're going to look at 3 different factors. We're going to look at some environmental conditions. We're going to look at the concept of cofactors and coenzymes. We're also going to look at some enzyme inhibitors. So let's look at environmental conditions. There's a few environmental conditions that will greatly affect enzymatic reactions. So for example, temperature. All enzymes are able to work optimally within a certain range. Now, high temperatures, because they're a protein, may denature the enzyme, meaning that it wouldn't work as effectively. Same with pH. Enzymes have an optimal pH range. If something falls to be too low in pH or too high in pH, it can greatly affect that enzyme's productivity, and same with ionic concentrations. So an excess of salt ions, for example, a very saline environment, can also affect how enzymes function. Okay, and remember homeostasis, one of its important jobs is to keep this within optimal ranges for proper enzyme activity. Cofactors and coenzymes, well, these are inorganic substances, so these are like our zinc and iron, and also our vitamins. And these are needed for proper enzyme activity. So our inorganic substances like zinc and iron, these are our cofactors. And our vitamins are our coenzymes. So for example, vitamin C is an important coenzyme for cellular respiration. We need zinc and iron for, while we need zinc for the immune system, and iron must be present in hemoglobin in order for it to pick up oxygen. So these are two very important factors that are added into our enzymes to make them work more efficiently sometimes. The last sort of thing that we'll look at is enzyme inhibitors. And within enzyme inhibitors, there are competitive inhibitors. Now, competitive inhibitors are chemicals that resemble the enzyme's normal substrate, and what they do is they compete for the active site. So this would be our competitive inhibitor, and notice that the region where the active sites bind are very similar. And in this case, they're going to compete with the enzyme. Now, they're not going to do the same function as the enzyme, and so as a result, that enzyme is not going to be breaking down the substrate as efficiently. Non-competitive inhibitors, these work a little bit differently in that they don't act on the active site, but what they will do is they act on another region on the enzyme. And when they bind to that enzyme, they change the shape of the enzyme, which alters the active site, and the substrate will not be allowed to bind to it. A lot of our poisons work very similar to this in that they change the shape of our enzymes or change the shape of enzymes in order to make enzyme activity very ineffective. So that concludes our podcast on enzymes, and I hope that was helpful.